All right, so we've got a whole stack of articles and notes you sent over. Looks like you want to do a deep dive on cryo-EM.
Yeah,
And not just what is IIS, but how it's changing the game for drug design.
absolutely. It's an exciting field.
Yeah.
We're talking about visualizing molecules at near atomic
It's
resolution.
amazing.
Yeah, just a decade or two ago, this level of detail was unimaginable.
It's like suddenly being able to see all those tiny little gears of a complex machine, that we only understood in abstract terms.
Exactly.
From your notes, it seems like you're particularly interested in how this level of detail
Mm-hmm.
translates into designing better, more precise drugs.
Yes, exactly. Traditionally, drug design involved a lot of trial and error.
Right.
You'd synthesize a bunch of different molecules, test them out, and see what stuck.
Just throw it at the wall and see what sticks.
Yeah, it was a bit like trying to pick a lock in the dark. But
Yeah.
with cryo-EM, it's as if someone suddenly turned on the lights, and now we can actually see the shape of the lock and the key.
Yeah. Okay. I'm intrigued. But before we get into the specifics of, drug design, let's break down what makes cryo-EM so revolutionary. Why is this such a big leap forward compared to older imaging techniques?
Yeah, so one of the biggest limitations of older techniques like X-ray crystallography was the need to crystallize the proteins you
Okay.
wanted to study, and many proteins, especially large, complex ones, simply refused to cooperate.
Right.
Imagine trying to take a clear picture of a hummingbird's wings mid-flight.
Oh, right.
It's just you'd end up with blurry images or nothing at all.
Yeah, nothing at all.
But cryo-EM bypasses that entire problem by flash freezing the samples. It's like hitting the pause button on those hummingbird wings, allowing us to capture their structure in exquisite detail.
Wow. So we're talking about seeing individual atoms within a molecule.
Essentially, yes.
Wow.
And the fact that we don't need to crystallize the samples means we can study a much wider range of proteins, including those that were previously considered undruggable.
That's incredible.
Yeah,
That's what I call a game changer. But it's not just about seeing static structures, is it?
right.
I remember from your notes something about capturing like these transient states and dynamic complexes.
Yes.
Those sound pretty important.
They are, and this is where cryo-EM truly shines. Proteins aren't just static entities. They're constantly in motion, interacting with each other, changing shapes and forming complexes. These transient states and dynamic interactions are crucial for understanding how biological processes actually work.
So it's like we've gone from looking at a still photograph to watching a high-definition movie of these molecules in action.
Exactly.
That must be giving us incredible insights.
It is. We can see how different parts of a protein move, how they interact with other molecules, and how these interactions change over time.
Hmm.
These insights are absolutely critical for designing drugs that can effectively modulate these complex processes.
So it's not just about knowing, the basic shape of a protein. It's about understanding its entire repertoire of movements and interactions.
Precisely. And with cryo-EM, we can actually visualize those movements and interactions, giving us a level of understanding that was simply impossible before.
This all sounds incredibly complex. I'm starting to see why those factoids you mentioned, visualizing molecules at near atomic resolution, uncovering receptor conformations, capturing transient states, are such a big deal.
Yeah, they really are transforming how we approach drug design.
Yeah,
And this leads us to the concept of precision therapeutics, which I believe is of particular interest to you.
it's fascinating. But I'm realizing that there's a lot to unpack before we get there, so help me connect the dots here. We can see these incredibly detailed images of molecules, even capture their dynamic interactions. But how does that translate to designing more effective drugs?
Right, so we'll think of it this way. Imagine you're trying to design a key for a lock, but you've never actually seen the lock. You might stumble upon something that works eventually,
Right.
but it's going to be a long and arduous process.
Yeah, you'd probably end up with a key that's clunky, doesn't fit quite right, maybe even damages the lock.
Exactly. But now imagine you have a blueprint of the lock with every groove and detail clearly visible.
Oh.
Suddenly, you can design a key that fits perfectly, unlocks smoothly, and doesn't cause any unintended damage. That's essentially what cryo-EM is doing for drug design.
So instead of, blindly testing thousands of molecules,
Mm.
we can use cryo-EM to identify the precise shape and dynamics of our target protein and then design drugs that bind specifically to it.
Yeah,
That makes a lot of sense.
and it goes beyond just designing new drugs.
Okay.
Cryo-EM can also help us understand why some drugs stop working. For instance, bacteria and viruses can mutate, and these mutations can alter the shape of their proteins. With cryo-EM, we can visualize these changes and understand how they lead to drug resistance.
So we can adapt our drug design strategies to stay one step ahead of these evolving pathogens. It's
Exactly.
like an arms race at the molecular level.
Precisely. And that brings us to another crucial aspect of cryo-EM's impact on drug design, the ability to visualize previously undruggable targets. There are many proteins in the body that play key roles in disease processes, but were considered undruggable because we couldn't visualize them clearly enough to design drugs that could interact with them.
And cryo-EM is changing all that.
It is. By revealing the structures of these elusive targets, cryo-EM is opening up whole new avenues for drug discovery. We can now start to explore these previously uncharted territories and develop therapies for diseases that were once considered untreatable.
That's incredible. It sounds like cryo-EM is really living up to the hype. But as with any powerful tool, I imagine there are also challenges and limitations.
Of course. One of the biggest challenges with cryo-EM is the sheer amount of data it generates. We're talking about terabytes of data from a single experiment. Processing and analyzing all that information requires specialized software and expertise. It's computationally intensive and can take a significant amount of time.
So it's not just about having the fancy microscope, it's about having the computational power and the expertise to make sense of the data it produces.
Exactly. And while the technology is constantly evolving and becoming more accessible, it's still relatively expensive, which can limit its availability to certain labs and research groups.
Right.
But even with these challenges, the impact of cryo-EM on drug discovery is undeniable. It's a true game changer.
For sure. I'm curious about specific examples where cryo-EM has been used to make, significant breakthroughs in drug design. Anything come to mind?
Absolutely. One of the most prominent examples is the development of COVID-19 vaccines. Scientists used cryo-EM to visualize the spike protein of the SARS-CoV-2 virus at an atomic level,
Wow.
which was crucial for understanding how the virus infects human cells and designing vaccines that could effectively block that interaction.
So cryo-EM played a key role in helping us combat a global pandemic.
It did. And that's just one example. Cryo-EM is being used to study a wide range of diseases, from cancer to Alzheimer's to HIV, leading to the development of more targeted and effective therapies.
So this all ties back to that concept of precision therapeutics we touched on earlier. It's like we're entering an era where drugs are being designed not just for a specific disease, but for a specific patient based on their unique molecular profile.
That's the ultimate goal. And while we're not quite there yet, cryo-EM is certainly pushing us in that direction. By providing such a detailed understanding of molecular interactions, it's paving the way for a future of personalized medicine where treatments are tailored to an individual's specific genetic and molecular makeup.
It's like science fiction becoming reality. I'm really curious to know what you think the future holds for drug design and development. Where do we go from here? What other innovations are on the horizon?
Well, I think cryo-EM is just the tip of the iceberg.
Correct.
It's a powerful tool, but it's not a standalone solution. The future of drug design is going to be about integrating multiple technologies and approaches.
So what else is out there? What other exciting, advancements should we be keeping an eye on?
One area that's really exploding right now is artificial intelligence and machine learning. These technologies are being used to analyze massive data sets of molecular information, identify potential drug targets, and even predict the effectiveness of new drugs.
So AI is becoming like a crucial partner in the drug discovery process.
Absolutely. It's helping us sift through mountains of data and identify patterns that would be impossible for humans to spot. And that's not all. Another incredibly exciting area is gene editing.
Wow.
Technologies like CRISPR are giving us unprecedented control over the genetic code, opening up new possibilities for treating genetic diseases, and even potentially enhancing human capabilities.
Gene editing. That sounds like something straight out of a science fiction movie. It's amazing to think about the potential, but, it also raises a lot of questions and ethical considerations.
You're right, and those are conversations we need to be having. But despite the challenges, I believe that the future of drug design is incredibly bright.
Yeah.
We're on the cusp of a new era of personalized medicine where treatments are tailored to an individual's unique genetic and molecular makeup.
It's like we're moving from a one-size-fits-all approach to a bespoke tailoring service for medicine.
Exactly.
That's a powerful image. But with all this talk about, cutting-edge technology, I can't help but wonder about the role of traditional methods. Will they become obsolete?
I don't think so. I see it as a synergistic relationship. Traditional methods like natural product chemistry and high-throughput screening will continue to play a vital role, providing valuable insights and complementing these newer technologies. It's about combining the best of both worlds to create safer, more effective, and more personalized therapies.
It sounds like we're on the verge of a truly transformative period in medicine.
Mm.
But as exciting as all this is, I think it's important to acknowledge that, drug discovery is still a long and challenging process.
Mhm.
What are some of the hurdles that researchers are facing today, and how might we overcome them?
You're absolutely right. One of the biggest hurdles is still cost. Developing new drugs is incredibly expensive and time-consuming, and the failure rate is still very high. We need to find ways to streamline the process, reduce costs, and improve our ability to predict which drug candidates are most likely to succeed.
So it's not just about coming up with brilliant ideas, it's about making those ideas a reality in a way that's both efficient and affordable.
Precisely. And that's where advancements in automation data analysis and even things like 3D printing are playing a crucial role. These technologies are helping to accelerate the drug discovery process, making it faster, cheaper, and more efficient. But there are also challenges beyond the technical aspects.
Like what?
Well, for one, we need to foster greater collaboration between academia, industry, and government. Breaking down silos and sharing knowledge and resources is essential if we want to accelerate the pace of discovery. We also need to address issues like regulatory hurdles and intellectual property rights. These are complex challenges, but I believe that by working together, we can find solutions that benefit everyone.
It's inspiring to see so much innovation happening in this field. It really feels like we're on the cusp of something truly remarkable. As we wrap up this deep dive into the world of cryo-EM and drug design, I want to bring it back to you, the listener. What's the one key takeaway you hope they walk away with?
That's a great question. I think the most important takeaway is that we're living in an incredibly exciting time for scientific discovery. The tools and technologies we have at our disposal are more powerful than ever before, and we're using them to unlock the secrets of life at an unprecedented pace. And while there are still many challenges to overcome, the future of medicine is full of promise. We're moving toward a world where diseases that were once considered incurable are becoming treatable, and where treatments are becoming increasingly personalized and effective.
It's a future worth striving for. And who knows, maybe one of you listening will be the one to make the next breakthrough discovery.
Keep learning, keep asking questions, and never stop exploring the fascinating world of science.
That's a perfect note to end on. Thanks for taking this deep dive with us. Until next time, stay curious.